Thank you and good morning. We very much appreciate the opportunity to present what's going on with AirPO at this year's OIS meeting. We are a public company, so I need to refer you to our forward-looking statement and all the SEC filings you'll find on our website. I wanted to start by giving you kind of a summary of where we are. Our lead program is in studying a small molecule AKB-9778 in the setting of diabetic retinopathy. I'd like to give you an overview of what we found a couple years ago in our phase 2A study called TIME-2. This was a three-arm trial of 3 months duration looking at AKB-9778 and Lucentis in combination, and DME and diabetic retinopathy. One arm received subcutaneous 9778 twice a day and sham intravitreal Lucentis injection once a month for 3 months. One arm received daily subcutaneous 9778 and intravitreal Lucentis once a month for 3 months. And then the other arm just received Lucentis and subcutaneous placebo for 3 months. Because of the design of the study, in that Lucentis was only given in the study eye, but 9778 is administered systemically, we have the opportunity of looking at effects in the study eye and also in the fellow eye. The results from that trial looking at diabetic retinopathy are shown in the bottom left-hand portion of the slide. We're looking at the fellow eye. We were able to show the same degree of two-step improvers in DRSS score compared to Lucentis. In combination was probably a little bit more effective. Importantly, in the fellow eye that only received 9778 or placebo, we were able to show a significant reduction, a significant improvement in the two-step improvement compared to placebo alone. Importantly, the unique thing about 9778 is that this is given systemically, thereby we can treat both eyes and also look at other potential tissues. The bottom right-hand corner shows the effects of renal function looked at 3 months after 3 months of duration of treatment, where we saw a roughly 20% improvement in UACR ratio in those patients that had some underlying degree of nephropathy compared to placebo, which had an increase in UACR ratio. So we were very excited about this. This all led into the current trial that we're conducting, a phase 2B study called TIME-2B of just daily 9778 in non-proliferative diabetic retinopathy patients, looking at the systemic exposure of this drug and to slow the progression of diabetic eye disease and possibly other diabetic complications like nephropathy. This study was begun in June of 2017. It's fully enrolled. It was a 3-arm study of once a day subcutaneous 9778, twice a day 9778, and placebo. These are all NPDR patients without DME or visual abnormalities. The primary endpoint is at 48 weeks, looking for two-step improvers. And the key secondary endpoints are looking at progression into DME and PDR, to progression of diabetic retinopathy, and specifically renal function. We upsized this study to 167 patients from 150, and we will have data early next year. In that trial, in the TIME-2 study, and a safety surveillance, we found something very interesting, which was that all patients who received 9778 after 3 months had a statistical significant reduction in their intraocular pressure compared to the placebo group. And the drop in pressure was 1 to 1.5 millimeters of mercury, but it was a bit surprising. When we looked at that data a little bit more carefully, we found that that patients who had high normal baseline intraocular pressure had a more significant drop in IOP of about 3 to 3.5 millimeters of mercury compared to those patients that had low intraocular pressure at baseline. There was no difference. And we saw a stat sig difference in all patients receiving 9778 at month 1, month 2, and month 3. So it really seemed to be that there was something going on here. And when you look at the evolving scientific literature, it kind of starts to make sense. Dr. Quagan's group designed a knockout mouse where angiopoietin-1 was reduced and Tie2 was inhibited, and that mouse led to a congenital glaucoma phenotype. Dr. Koh's group did something a little bit more elegant where After the mice were born, they had a fully formed Schlemm's canal. Knocking out Tie2 and angiopoietin-1 led to intraocular pressure and a reduced retinal neural layer. And the evolving human genetics now is showing there's a correlation of angiopoietin-1 and Tie2 mutations associated with congenital glaucoma in babies and with increased finding in primary open-angle glaucoma. So when we look at the whole scheme and the body of evidence, as demonstrated in Dr. Koh's paper, Tie2 really appears to be associated with a healthy Schlemm's canal lumen, intact giant vesicles, and appropriate aqueous humor outflow. As mice age and Tie2 is inhibited, you get a glaucomatous phenotype where you have disrupted Schlemm's canal lumen, decreased giant vesicles, and so forth. So there really does appear to be a correlation of Tie2 and glaucoma. So what we did is we then developed a topical ocular dose form of AKB-9778 and studied it in the normotensive New Zealand rabbits. On the left-hand part of this graph, we gave these bunnies 10 milligrams subcutaneous BID, just as we did in the Time 2 study, and it replicated about a 1 to 1.5 millimeter drop in IOP, which was sustained for 24 hours at the end of treatment. When we developed a low concentration, low dose topical dose form, we also saw a drop in IOP, but it was not sustainable. However, when we made a higher concentration, high dose topical dose form, we saw again a more pronounced drop in IOP, which did appear to be sustainable for 24 hours. So with this information from both our clinical data and our non-clinical data, we believe that activation of the Tie2 pathway via AKB-9778 inhibition of the phosphatase reduces intraocular pressure in humans and in rabbits, and the proposed mechanism is via the Tie2 activation mechanism. We are developing a topical dose form, which will be going into clinical development early next year. Thank you very much.